So I've stepped out of the lab to explain to you that science doesn't always take place in our science lab. We're talking about bacteria, and I want you to know that there is bacteria on everything. That's why I came out to this lobby here, because every surface on the planet is covered with bacteria. Inside of you, you are full of bacteria. Bacteria is not always bad. It can be. There are some examples in which it is, and I'll explain those later. But I want you to know that bacteria is just on every surface that we touch. And normally we would, in order to see bacteria, we would do an experiment inside the lab. Let me explain to you what that's like. So here's how this experiment works. I've got two cameras set up, one straight out here, one over here, so that you can see all angles. In order to go collect bacterial samples, what I would give you is a special Petri dish. This Petri dish would have food at the bottom of it called agar. That agar is going to give our bacteria the best chances to get a source of energy. What else do they need? Well, inside that agar is some water, and every living thing needs water. As well as we're going to give it the best possibilities. We're going to nurture this. We're going to keep it nice and warm underneath our heat lamps in order to give it the best opportunity to grow. So what I would normally have you do is take a cotton swab. You would swab something, get all the bacteria that's on there, because bacteria is on everything. We cover it up, we come back, we wipe it on our dish, cover it up, and put it inside our incubator, place it there, get nice and warm, and then after so many days, we would come back to this and we would open this up. And when we open it up a couple days later, there's a couple things you'd notice. One, there's definitely a stink to it. There is a smell because all living things give off or eliminate waste. And some of the things that the bacteria would be eliminating don't smell so good. So we would take a look at our sample, pull it out of here, and it might be, oh, coarse focus knob. Oh, I can see that. We don't want to open this up. We don't want to expose ourselves to any bacteria. But we would take our stereoscopes that we learned about. We would place our sample underneath that stereoscope, and then we would examine what that bacteria looks like. All right, my little human beings. So if I were to send you around the school to sample things, what would you like to sample? I mean, in the past, we've had things like door handles, lockers, library books, gym equipment, gym equipment, and the gym, parts of the cafeteria, the checkout area and the line, and always toilets. So what would you swab if you had the opportunity? I would like to hear your responses and explain why you would swab that area for bacteria. Remember, everything has bacteria on it, but the amount of bacteria is kind of what we would look for when doing this experiment. So my question to you is, what would you swab around the school and why? Think about every surface that is in a school building. Which one would you want to swab? And then explain why you would want to do that. You've always got to have a reason why when sciencing. So give an explanation. You're not going to be able to see your classmates' answers until you give your own answer.